Today I'd like to tell you about the geroprotective effects of the antiretroviral drug Zidovudine and how it extends healthy longevity via ATF4. So I certainly don't have to tell this audience that there's a strong genetic contribution to longevity and that there are known genes that can prolong life and therefore delay disease incidence. And one of those main genes most best known is FOXO3. So FOXO3 has been shown its overexpression or activation can extend longevity across the phylogenetic tree, and even human genome-wide association studies have shown associations of longevity in humans. So the main point of this project was to see if we could identify novel geroprotective compounds by mimicking the transcriptional effects of FOXO3. And we did that using the Library of Integrated Network-Based Cellular Signatures, or the LINCS database, which is basically this huge repository of transcriptional data, cell lines that have different genetic perturbations, and also a lot of drug interventions. So we utilized their transcriptome for FOXO3 overexpression, and we overlaid it with the nearly 3,000 candidate drugs that are in that database. And then we ranked and scored them for how well those overlapped. We subsetted that list for drugs that were already FDA approved, and we ended up with this top 5 list of compounds. We were quite pleased to see that our top 2 hits were already known to extend lifespan, so ciproheptadine and, of course, rapamycin is one of the best known longevity interventions, as well as a few drugs that are less known or unknown in the longevity space, which are atracurium, sildenafil, and zidovudine. So the first thing we wanted to do is make sure we could validate the transcriptional effects that we saw in our in silico drug screen. And so we treated HL1 mouse cardiomyocytes with these different compounds, and we tested for expression of FOXO3 as well as some of its downstream targets. And indeed, we saw that there were activation of the genes that we expected here. So the next step was to see if we were going to see geroprotective effects, and for then we moved to our favorite model organism, the C. elegans. And so at this point, we dropped ciproheptadine and rapamycin from the screen because we knew they extended lifespan, but we tested sildenafil, zidovudine, and atracurium. In our hands, at the concentrations we tested, sildenafil did not extend lifespan, whereas zidovudine and atracurium robustly and reproducibly extended lifespan in the wild-type worms. Our next question was about the mechanism, and since we were seeing this FOXO3-like transcriptome as well as some lifespan extension, we figured perhaps we were seeing FOXO3 or in the worms DAF16 activation. So we tested these compounds in a DAF16 mutant. And interestingly, these 2 compounds diverged here. So while zidovudine was still able to robustly extend lifespan in the DAF16 mutant, atracurium, the lifespan extension was completely ablated. So atracurium, these data are already published, so I'll run through the mechanism very quickly. Ultimately, we were able to show that atracurium, which is a muscle relaxant compound, it inhibits the neuromuscular junctions acetylcholine receptor via a subunit called UNC38 in the worms, and that this causes nuclear localization of DAF16 and then downstream effects that cause the longevity. But the main point of my talk today is this drug Zidovudine, which we found particularly interesting because it's activating this FOXO3-like transcriptome, but not via FOXO3 or DAF16. So I'll give a little background on what Zidovudine actually is. So it's a nucleoside reverse transcriptase inhibitor, or NRTI. It's also known as azetothymidine, or AZT, and because it's a thymidine basically with an azide group attached, it's been shown to additionally incorporate into the nuclear mitochondrial DNA of the host organism as well. And because of that, when it was first given to HIV patients, and it was really the original HIV treatment drug, patients experienced enormous side effects, and in the end, most of those have been attributed to mitochondrial toxicity because this compound can also inhibit polymerase gamma. So we were quite interested about why we were seeing these longevity effects in the worms, so I'll jump back. I've already shown you that zidovudine can extend lifespan, but this is a health span assay as well, so this is worm mobility, how fast they can move across the plate. And you can see that at day 1, the worms actually have slightly reduced mobility upon treatment, so this is after 24 hours of treatment. But later in life, the worms treated with zidovudine have significantly more mobility than their DMSO-treated counterparts. We were very interested in the mechanism here, so we performed RNA sequencing. And while we saw a lot of interesting changes, what really jumped out at us when we performed this GO-term cluster analysis, you can see, is that we saw downregulation of GO-terms related to translation, as well as peptidase inhibition, in addition to GO-terms related to proteolysis were up. And so this suggested to us that we were seeing something really significant happening in proteostasis. So based on that, we performed a sort of hand-picked small screen to see if we could identify a regulator involved in these processes. We chose these different genes based on both our lab's experience with proteostasis relation to lifespan, as well as some suggestions from literature. And as you can see in the bottom right-hand corner, that mutant of ATF4 was the only mutant that prevented the lifespan extension we saw with this compound. ATF4 is highly involved in the integrated stress response, and it's also been shown to be a major regulator of mitochondrial stress. So that sort of fits with this mitochondrial toxicity we've seen previously with zidovudine. ATF4 was also particularly interesting to us because my colleagues in Riekels's lab were recently able to show that there is a mitochondrial and cytosolic translational balance. And if you perturb that by reducing mitochondrial translation, something that was very well described this morning by Johan Aurks, you can inhibit mitochondrial translation, which then inhibits cytosolic translation and induces longevity. And both of these factors are also dependent on ATF4. So with that in mind, we actually returned to our RNA sequencing data to see if we could see a similar effect with the ribosomal genes in these worms. And indeed, almost all cytosolic and mitochondrial ribosomal genes were downregulated when worms were treated with zidovudine. In addition, we performed metabolomics in these worms. And I'll save you a lot of that data, but we checked amino acid abundance kind of in relation to this potential downregulation of translation. And we saw increased amino acid abundance in these worms. I also returned to the worms with inhibited mitochondrial translation, and they follow a very similar pattern. So together, these data suggested to us that we were seeing potentially downregulation of translation, at least in transcriptional and metabolic effects. And actually, last night, we received data from our collaborators that demonstrates that indeed we're seeing reduced translation, which we tested via polysome profiling. But unfortunately, I didn't have time to include that here. So last but not least, we wanted to see if this effect was exclusive to zidovudine or if other NRTIs had similar longevity effects. So we tested a full panel of NRTIs in these wild-type C. elegans, and the only drug that was able to robustly extend lifespan is called Didanosine, and this is an adenosine analog. But you can see here as well that it actually took 4 times the concentration of Didanosine to reach about half the median lifespan extension that we observed with zidovudine. When we moved Didanosine also to an ATF4 mutant worm, indeed the lifespan extension was ablated. So despite the fact that zidovudine is probably the more potent compound, they seem to act via the same mechanism. So I'll conclude for the talk as a whole that our in silico drug screen, where we mimic the transcriptional effects of a known genetic longevity intervention of FOXO3 overexpression, was able to identify novel geroprotective compounds in atracurium and zidovudine. When we probed these mechanisms, we were able to identify that atracurium inhibits the acetylcholine receptor at the neuromuscular junction via the subunit UNC38, and this causes nuclear localization of DAF16 and downstream effects that increase lifespan. And lastly, that the NRTIs zidovudine and didanosine extend longevity in the worm via this mitochondrial stress regulator of ATF4, and likely, and I would like to say now, via reduced translation. I'd like to thank everybody who was involved in these projects, particularly my supervisors, Riekels and George, as well as my collaborators both inside and outside the AUMC, and of course, you all for your attention. Thanks.
Thank you so much, Rebecca. Very exciting stuff. Do we have any questions? We have several. There's one down there.
Well, let's talk. So do you think the drug effect is caused by the uptake of the worm itself or caused by bacterial metabolism?
So we did actually test to see if we performed lifespans with killed bacteria to see if that was playing a role. And in fact, the lifespan extension remains, so it's not bacterial metabolism of the drug.
Thanks for the great talk. I was wondering if you checked about function, like mTOR function and some of the traditional mechanisms of increasing activity of FOXO, if that was different under zidovudine.
So we didn't test necessarily an mTOR mutant in these. I did look at some mTOR-related gene expression in the RNA sequencing data and didn't see massive differences, but of course it's a highly similar pathway, so it would be interesting to look into it further.
All right. Well, thank you so much, Rebecca. Fantastic work. And I want to thank all of the young presenters to be on time. That's amazing. We're even a few minutes ahead. All the old folks could certainly learn something here. So we will have a break. There are posters outside. Remember to go upstairs. It's good for you. And it's good for the people upstairs who are waiting for you eagerly.